Bio 181, Chapter 12. We're looking at genetics and inheritance, and we're going to look at Punnett squares. But first, we got to talk about how Punnett squares work, and they really work because of the random segregation of the genetic material during meiosis. So the principle of segregation, that is, you have two alleles for a given gene and they segregate during gamete formation. So in the formation of the sperm or the formation of the eggs, right, those alleles are going to segregate, and that process is random. Okay, so that's important for determining a method to model it. It's going to be probability, sort of like flipping a coin. So during fertilization, sorry, so we've got two cells here. I'm just going to say, let me model this. So here's one cell, here's one parent, and they've got, let's just do one chromosome over here, and it's got an allele, say capital A here, and a lowercase a over here. Okay. When this goes through meiosis and you end up with 4 daughter cells, okay, this would have actually been, replicated into sister chromatid, like this before segregation. You would have 4 daughter cells, and 2 of them would have the capital A and 2 of them would have the lowercase a, okay? So say this is the egg that was formed, and then over here, if you had the same thing, right? This parent, oh, let's just say, whoops, I'm trying to draw 2 here. So let's say in this case, we had, yeah, the same thing, capital A over here and lowercase a over here. Okay. So then the sperm, there's going to be 4 sperm that are formed, 2, 4, okay. And again, they either have the capital A or the lowercase a, the recessive allele. And these are going to, at random, meet, okay? That's a random process of fertilization, pretty much so. I mean, we're going to say it's random. And then it's going to create the zygote, which is the next generation. So in this case, it would be 2 little a's if these 2 happen to join. Oops, did I skip one? Okay. Independent assortment, this was really covered when we talked about meiosis, but it is about the fact of the segregation of these pairs of chromosomes are independent of each other. So depending on how they line up initially, you're going to have different final possibilities. So in this case, you can see one parent shown in blue, both of the chromosomes from that parent over here on the left, both of the parents in red on the right, and that's going to result in the genetic arrangement shown in the top. Whereas equally likely on the bottom, you could have, one parent has, for the blue parent chromosome on the left, one of them, and then the red also on the left. That would result in this arrangement here. So just by how they're lining up in the center to begin with, you're going to have a lot of different options. So that is also a random process. Okay, quick terminology, generations, that is, the individuals in the sequential line, parent and offspring. P is generally for parent. F1 is for the first filial generation. So the parents' children, and this would be the grandchildren, right? So that is basic terminology. We're going to be modeling this with a Punnett square. So we use the science of probability. That's a field of mathematics or statistics that looks at how, if we know the genetics of the parent and we know it's a random process to create the offspring, how can we model it? How common is it going to be to get X genes in an individual? So that's where we use a Punnett square. A Punnett square is a mechanism to diagram all of the possibilities in a fertilization event. So what happens is along one side, so you'll put, say, let's just go with the male across the top. Doesn't always have to be that way. We're going to put the female over here on the side. So on the top, if we're modeling a sperm, right, the sperm is going to bring in, if we're just looking for one trait, and say the parents had one of each, they had a dominant and a recessive trait, okay? Then you would put that along the side for the parent. So there's the male. Now, let's say the female, maybe she was homozygous dominant, right? She only had the dominant allele. So both of her eggs would have that same allele. So we put all of the possibilities for the gametes, which are those haploid cells that are produced as a product of meiosis, on one side of the square, and then from the other parent on the other side. Then we model fertilization, the sperm and the egg meeting. They combine and form the zygote. So the zygote here in this case is going to be homozygous dominant. This zygote that's formed from this sperm, right, and this egg is going to be heterozygous. This one is also going to be homozygous dominant, whereas this one again is heterozygous. So then you can count up, okay, out of 4, we have 2 out of 4, or 1/2 of the possibility for the offspring are going to be homozygous dominant. And then similarly, 2 out of 4, or 1/2 of the offspring are going to be heterozygous, okay? So that's a probability of 0.5 for homozygous dominant, a probability of 0.5 for heterozygous for this example. If we were to talk about the phenotype, now remember the phenotype is the physical expression. So let's go back and say capital A is for black fur and lowercase a is for white fur. In this case, the phenotype, you would have 100%. I shouldn't go to percent. I should go 4 out of 4 to be consistent. So 4 out of 4, or 1, okay, are all going to be black, right? So out of a total possibility of 1, which would be 100%, they're all black. So probability is a number between 0 and 1 by definition. So probability is from 0 to 1, whereas percent, which we're usually a little more familiar with, is a number from 0 to 100. So probability of 0 is 0, probability of 1 is 100%. Okay. So in this case, we've got 50% probability of having homozygous dominant offspring. We've got 50% probability of having heterozygous, and we've got 100% probability that the offspring is going to have black fur. Okay. A specific example that you need to know is called a monohybrid cross, and this is for the F2 generation. And so we've had a few crosses before this, but this is an important one. So the main thing is we have one trait that we're looking at. So that's where this word mono comes in. That just means we're looking at one trait and not a whole bunch. So we're just looking at fur color right now. We're not looking at fur color and eye color and height, right? So monohybrid. The hybrid, we're hybridizing. So we're crossing 2 parents. And you can think of it like plants, which Mendel initially, who was the original geneticist to formulate the theory, worked with plants a lot. So we're starting with a heterozygous parent in both parents. So that means we would have a capital, meaning a dominant allele, and a lowercase, meaning a recessive allele for each parent. So both the male and the female parent have the same genotype. So we're going to write that along the side. So again, I'm just going to pretend This is sperm up here, just so I really want you to get that that's what we're modeling. So this is the possibilities for the genes that are carried in the sperm along the top. The genes that are carried along the side, the allele for those genes in the egg are here, and that would, again, in a heterozygous parent, you have one dominant, one recessive. Okay. So you fertilize, you just put the letters, above and on the side together. So that's what you do for each box. Okay. Okay. So in this case, we have out of 4, right? We have one that's homozygous dominant. We have 2 that are heterozygous. So both of these, okay, are heterozygous. Heterozygous. And we have 1 that is homozygous recessive. Recessive. So the probability for each of these, you can calculate, this is 1 out of 4. For these 2, this one is 1 out of 4, plus this one is 1 out of 4. So together, that's, you would add them up over here. I'm just going to write the heterozygote is going to be 1/4 plus 1/4, which is 1/2, right? We already said the homozygous dominant was 1/4. And then we have the homozygous recessive, which is 1/4. Okay. So that would be 0.25, 0.5, 0.2. So those are the genotypes. This is the genotypic ratio right here. Okay. The phenotypic ratio, which is always something that is mentioned when we talk about monohybrid crosses, is the ratio of the dominant phenotype to the recessive phenotype. And so in this case, both the homozygous dominant and the heterozygous have the dominant phenotype. So the dominant is going to be 3 out of 4, 0.25 plus 0.5, right? That's 0.75 probability, or 3 out of the 4 squares. So the ratio is 3 with that dominant allele to 1 recessive allele. So that's a 3-to-1 ratio. Again, here's my dominant. This is my dominant phenotype. Okay, so if the dominant phenotype was black, these would all be black. Okay, and if the recessive phenotype was white, then this would be white here. Okay, so you've got 3 black phenotypes, 1, okay, 2, 3, right? That's this 3 for every 1 white phenotype here. All right. Okay, the last one, I'm not going to diagram this whole thing out. It's a dihybrid cross. So now we're talking about 2 traits. We're still talking about heterozygous parents. So both of the parents are heterozygous in both traits. So let's look here. So the example here for the parent, the genotype for the parent, right? We said it's heterozygous in both traits. So what that would look like in this case is they have 1 dominant and 1 recessive allele for the first trait, which in this case is represented with the letter Y. And then they have a second trait. So we're adding in another trait. And now we're going to use a different letter. They have 1 dominant allele and 1 recessive allele for that trait. Okay. So their genotype is now 4 alleles when we're talking about 2 traits. In this case here, this is the Y. It's going to represent the color of the seed. So this is the color. And the R is going to represent the shape. So the color, capital Y is yellow. Wait, is that right? Yeah, capital Y is yellow, lowercase y is green. For the shape, the capital R is round, right? Yeah, round. And the lowercase r, the recessive allele, is wrinkled. So if you look at the genotype, it's heterozygous in both traits. So what's the phenotype? What's the physical expression? Right? So the phenotype is going to be the dominant phenotype for both, right? Because as long as it has the dominant allele, it's going to express it. So that means both of the parents are going to be yellow and round. Okay, yellow and round. And that's what's shown here. This is shown that this is the generation that is going to be bred together to create our F2 generation for the dihybrid cross. If we were to do this, when we separate these, now instead of just having one allele in each gamete, we're talking about two traits. So in each of the sperm, right, you're going to be carrying an allele that codes for color and an allele that codes for shape. So we've got two listed at the top instead of one, as we did in the monohybrid cross. Okay. And then we have the same thing for the eggs over here on the other side. And when they create the zygote, which is the cell that arises, that single cell after fertilization, it's now going to have 4 alleles in each cell. Now, it's 4 alleles of interest, right? We're only talking about traits that we're interested in, and that's what we're modeling. So in this case, you can see that all of the alleles are listed. There's 16 boxes. And you can see the genotypes, those are the letters. You can see the phenotypes, those are the colors and the shape of the seed, right? In this case. For the phenotypic ratio, in this case, we've got 9 out of the 16 that have both dominant traits. So these are both dominant. So they look like the parents, okay? Both of the dominant phenotypes. So they're 9 out of 16 are yellow and round. 3 out of the 16 are, well, they have now the recessive phenotype of being green, but the dominant genotype of being round, right? So it's 1 dominant and 1 recessive, okay, phenotype. And then, oh, yeah, so that's 3. Then there's another one, which again has 1 recessive phenotype and 1 dominant phenotype, okay? And then there's a single one out of the 16 that has 2 recessive phenotypes, okay? So this, in this case, whoops, they're both recessive. So 1 out of 16 offspring is going to be wrinkled and green, right? All right, that is it for this video.